Hello everybody, and welcome to this video on reaction mechanisms, where here we're going to introduce some of the fundamental concepts, some of the fundamental terminology we're going to need to be more and more familiar with to understand reaction mechanisms from the perspective of organic chemistry. Okay, so let's start by reminding ourselves what we've learned about reaction mechanisms from our general chemistry or AP chemistry courses. We've learned a little bit about the idea associated with things like rate laws, transition states, how to figure out whether a reaction has intermediates, the activation energy of a reaction, the elementary steps. So these concepts here we've learned a little bit about already in AP chemistry. So for example, a reaction might have many elementary steps, each of which is represented by a hill in the reaction coordinate diagram. And at moments that are at the top of the hills, we have things called transition states. And at times in between the tops of the hills here in the valleys, we have intermediates, right? Intermediates are species that were not there to start. They show up at some point, they go away as we work our way from reactants to products. And there are kinetic issues we talked about, right? The height of the tallest hill is associated with the activation energy of a reaction. There are thermodynamic considerations, right? What's the difference between the energy of where the reaction started to the energy of where the reaction ended up? And that would be something like the change in the free energy of the reaction. And that could tell us if the reaction were spontaneous or non-spontaneous. So from general chemistry or AP chemistry, we've touched on a lot of the fundamentals of reaction mechanisms. What we're going to do here in organic chemistry is we're going to take many of these same ideas and we're going to adapt them to the setting of organic chemistry, but along the way, we're going to need to develop some new terminology that we haven't bumped into before. And we're also going to combine these ideas with some of the electron arrow pushing that we've begun to do, that we first started to do with drawing resonance structures, and we've started to do with things like acid and base reactions. We're going to bring in more arrow pushing processes that help us show how reaction mechanisms occur, how reactions occur within the context of organic chemistry. Okay, so let's get to some of the new terminology we're going to have to be comfortable with as we delve into organic chemistry reaction mechanisms. So there are two kinds of molecules that will react with each other in an organic chemistry reaction. One of the partners in that process is called a nucleophile. And what a nucleophile is it's a molecule that contains an atom with high electron density. These things are the nucleophiles. Now we could sort of break this word down into its root parts. Nucleo meaning the nucleus. Phile, I think, is a Greek root for the word love. So a nucleophile is something that's going to want to join up with, it's going to want to make connections with other atoms. So nucleophiles are in search of other atoms. And the reason they're in search of other atoms is the nucleophile itself contains a region, or maybe even has an actual charge, that involves high electron density. So it might have an actual negative charge, which would tell us it has a lot of electron density, or maybe it's just polarized in a way that shows that there's a region of high electron density. So for an example of something that's polarized, we can take a look at this molecule here on the left, okay? This lithium CH3 molecule. Now, lithium is less electronegative than carbon. So the lithium-carbon bond is actually polarized in such a way that the carbon has a noticeably pseudo-negative charge on it. So this carbon right there is going to be acting as a nucleophile. It has a region of high electron density, and it's looking to share that electron density with some other organic molecule. Then we have something like the molecule ethoxide, which has an actual honest-to-goodness a coulombic negative charge on the oxygen. So this oxygen is a region of high electron density, so high in fact that it's got a negative charge. And so this negative electron would be looking to do a reaction and have its electron density head towards something that has a positive charge, maybe, for example, an H+. So the ethoxide oxygen is a nucleophile that is looking to share its electron density with something that's lacking electron density. The same can also be said of the lone pair electrons on ethanol. Now, while ethanol has an extra hydrogen, so it's neutral, it doesn't have a negative charge like the ethoxide does, these lone pairs on the oxygen are regions of high electron density, and they too can act as nucleophiles. The double bond of an alkene is also a region within a molecule of relatively high electron density. So this area of electron density, this double bond, can also go in search of something that has a lack of electron density. It too could maybe go in search of something like an H+. So one half of many kinds of organic chemistry reactions is this nucleophile. It's something that has extra electron density that is looking to be shared with another molecule that is lacking electron density. Now, you may have noticed that this term here, nucleophile, we've kind of bumped into a term that already shares this property, a region of high electron density. And so nucleophile is kind of really just a fancy word for what we've referred to before as a Lewis base. Okay, a Lewis base. You might think of the molecule NH3 is the classic example of a Lewis base that you bump into in general chemistry. Because of the lone pair of electrons there, it is acting as a Lewis base. It can go ahead and add those electrons to something else that is missing or is low on electron density. So that's our first new term that we have to get comfortable with, nucleophile. Okay, something that is willing to share its electron density with something else. All right, what will it share its electron density with? It will share its electron density with an electrophile. So molecules that contain atoms with low electron density, these are the electrophiles. All right. So these are the molecules or portions of a molecule that will be receiving the electron density from the nucleophile. So maybe as a clear example, carbocations, right? This carbon in the center of this molecule, that carbon has a positive charge on it. So it is clearly lacking electron density. So then, a nucleophile, I'll just abbreviate a nucleophile as NUC, maybe with a lone pair of electrons, that nucleophile will go after that electrophile. So the high electron density of the nucleophile goes after the low electron density of the electrophile. Now that's a case of where there's an overt positive charge on a molecule. We could, like we had with nucleophiles, we could have a polar bond. And here's a carbon-chlorine bond that is going to be a polar bond. So the carbon of the carbon-chlorine bond, that is missing electron density due to the electronegativity differences. So we could have a case where a nucleophile with its high electron density might go after the pseudo-positive, partially positive end of a polar bond, which is a site of an electrophile. So we have this partnership. The high electron density nucleophile will want to join up, will want to attack, quote unquote, the low electron density electrophile. And so here, the word electrophile is really just fancy for the term Lewis acid, okay? Lewis acid. And so something is a Lewis acid if it is lacking electron density. Okay, so many reactions in organic chemistry can be represented as a nucleophile, quote unquote, attacking an electrophile. We'll have this nucleophile-electrophile pairing. Now, this is just kind of general terminology. What specifically is the nucleophile? What specifically is the electrophile? What specifically are they doing with each other? Well, those are the details that we will get into as we look at specific kinds of organic chemistry reactions. All right, so just to add to some of the terminology here, so a portion of a molecule that can react like an electrophile or a Lewis acid is also referred to as the electrophilic center. Okay, so if there is part of a molecule that is willing to gain electron density, we would call that part of the molecule the electrophilic center. The portion of a molecule that can react like a nucleophile is going to be called a nucleophilic center. Okay, so if there's part of a molecule that has high electron density that could possibly be shared with a different molecule, then that part of the molecule is called a nucleophilic center. Okay. All right, so to get a feel for regions of a molecule that might act as a nucleophile because they have extra electron density, or might act as an electrophile because they're lacking electron density, let's take a look at this example of a fairly long and complicated molecule that I've drawn here. Now, this molecule may not actually exist in the real world. We're just kind of using this as an example. And so what I'm going to do is I will circle in blue any part of the molecule that could act as a nucleophile. All right, so any part of the molecule that has high electron density. So maybe you might want to pause the video right here and just think about, okay, which parts of this molecule could be nucleophilic centers, could act as regions of high electron density. All right, so maybe pause the video and think about it for yourself, and I'll start to answer it in a moment. So which parts of the molecule could act as nucleophiles? Well, the oxygen with the negative charge could act as a nucleophile. It's clearly got a region of high electron density. The lone pairs on the nitrogen here of this amine group, that could make that nitrogen act as a nucleophile. All right, the lone pairs on the oxygen of this alcohol could cause it to act as a nucleophile. So too could the lone pair of the oxygen here. Right, this carbon-oxygen bond is polarized in the direction of the oxygen. So the oxygen here could act as a nucleophile. There's one other spot of the molecule that could also act as a nucleophile. We have lone pairs on this oxygen. Those lone pairs could be pushed in and cause the electrons in the double bond to be pushed out. And so we would end up having a negative charge on the carbon. So the carbon on the end of that double bond could also be a region of high electron density, could also act as a nucleophile, as well as just the double bond in general, that could too act as a nucleophile. So those are the regions of the molecule that are going to tend to have high electron density. And so therefore, they're spots that could be looking to add to an electrophile. So now let's look at the other end of the spectrum here for the molecule. Which parts of this molecule could be an electrophilic center? Which parts could look like they would want to gain or have electron density added to them? Well, one region that I think is pretty obvious is probably this positive oxygen, right? So that part of the molecule could be looking to have some electron density added to it. Now also, because of the possibility of polarity here in the carbon-oxygen bond, the carbon of the C double bond O is also a region that could be considered electrophilic, as would this carbon on the other carbon-oxygen double bond. That carbon would also be electrophilic, so would be lacking in general in some electron density. Okay, so we're only going to look at a couple of reactions where nucleophiles are reacting with electrophiles as we go through the rest of Organic Chemistry 1. Some of the other types of nucleophile electrophile reactions we will get into in Organic Chemistry 2. But you should begin to get comfortable with, okay, which portion of this molecule has high electron density and might therefore be nucleophilic? Which part of this molecule might have low electron density and therefore might be electrophilic? Okay. All right, so this starts then to the possibility of us looking into some of the details, some of the actual arrow pushing that we could do to start to articulate organic chemistry reaction mechanisms. So I want to start bringing in the list of common patterns that we see in organic chemistry reactions. Now you might remember we had the 5 patterns of resonance for organic chemistry, right? Things like the allylic positive charge, the allylic lone pair, the conjugated double bonds in an aromatic, right? We could move those around. So we had our 5 patterns of resonance. Well, when it comes to reaction mechanisms, here's the cool thing. All reactions in organic chemistry that we're going to see, all reaction mechanisms, can be broken down to being one of 4 different main ways that we can move electrons in organic chemistry reaction mechanisms. There are 4 patterns, we will call them 4 patterns, 4 moves of organic chemistry reaction mechanisms. And I'm going to go ahead and list those 4 here. The first one we will call nucleophilic attack. Okay, so that is something that's going to create a bond between a nucleophilic center and a receiving electrophile. Another pattern that we're going to see is something called loss of a leaving group. Oftentimes, an atom or a group of atoms, more likely, needs to kind of fall off of a substrate in an organic chemistry reaction. So we'll see this pattern called loss of a leaving group. This third pattern is something we've already spent some time talking about, and that is the simple issue of a proton transfer. We have already talked about how to draw the arrows for a fairly simple Bronsted-Lowry acid-base reaction, but that's essentially what we would be talking about there. So proton transfer. And then the last common type of reaction mechanism move we can make in organic chemistry would be what we will call a rearrangement. Now, there are different kinds of rearrangements that happen in organic chemistry. We'll be looking at something called a hydride transfer or a methyl transfer in detail here during this class. And there are other kinds of rearrangements, but these are in general the 4 patterns that we're going to see. Every move we make in an organic chemistry reaction mechanism, we will be able to catalog as one of these 4 moves. Okay. All right. I think I'll end the video there just to kind of get the feel for what direction we're headed. And as we proceed in class down the line, we'll get into the nitty-gritty details of these 4 moves of different patterns of organic chemistry reaction mechanisms. All right. I will see you in class soon.